Hello, hello Charchika, Suchita, Kapil. Hi. If I am audible and visible, please say hi in the comment or in live chat if I am audible and visible. Okay, guys. So we'll start with today's session. It will be a short session about hybridoma technology. Okay. So let's crack UPSC CSE with India's largest learning platform, that is Unacademy. Okay, guys. This is all about me. You know about me already. Fine. There is a special video which talks about me. Then on Unacademy, you will get regular daily live classes, which is very important. And all the students who are regularly attending my classes, you guys know this, that you get access to daily live classes. Then Unacademy also provides you structured courses, and as well as there are regular live test and quiz till date on Unacademy learning platform. We are going through previous year question paper in test format, and ultimately all these daily live classes will provide you unlimited access of recorded videos. India's top educators will educate you on Unacademy. You can see the list. And on Unacademy, courses are available from polity to science and technology, and from governance to interview preparation, as well as answer writing for mains examination. There are many options to subscribe Unacademy from 1 month to 24 month. I suggest you to go for 12-month subscription. The fees is 40,000 rupees. Use my code SKUS and you will get immediate 10% discount. Okay, and you will pay 36,000 rupees. So please subscribe to Unacademy learning platform. If you are opting for a 24-month subscription, use my code SKUS and you will get 10% discount, and your fees will be 43,200 rupees. So don't forget my code SKUS. Okay, fine. Now we are starting with the actual topic. The name of the topic is Hybridoma Technology. Now if you can hear me, and good afternoon everybody. You can hear me as well as you can I'm audible and visible now, right? I'm audible and visible, guys. Yes, fine. So you guys will answer me where exactly we mentioned hybridoma technology. Fine. So I will start with first slide. It is purely for factual information, so not much important. First, this technology was invented or developed by these two scientists, George J.F. Kohler and Caesar Milstein. Okay, and for that they also got Nobel Prize in 1984. Now here I already told you hybridoma means hybrid cell. In this technology we are using hybrid cells, that's why this technology is known as hybridoma technology. So first keyword or first statement that you need to remember is hybrid cell. Now you will ask what is a hybrid cell, that you will learn in next few slides. So what we do in hybridoma technology, we use hybrid cell. That's why the name hybridoma technology. Okay. Second, these hybrid cells, they produce monoclonal antibodies. These hybrid cells, they produce monoclonal antibodies. Now you already know about monoclonal antibodies, and again we will learn about monoclonal antibodies in these slides. So this is about hybridoma technology basics. Yeah, basic introduction of hybridoma technology. Okay, Neha Yadav, good afternoon. Now see, one by one, huh? Just remember each and every fact. This is basic as well as current affair, and it is important for your UPSC prelims 2020. Those who are appearing for 2020, for them it is very important, and those who are appearing for 21, your basics are being covered through these current affairs. Okay. Now the hybrid cell that we talked about in the first slide. Okay, this hybrid cell has the capacity of antibody production that we already discussed. And what type of this antibody is? This is a monoclonal antibody, right? Here you need to remember first cell. See हम क्या कह रहे हैं? Hybrid cell, right? Good afternoon Mayank. We are saying hybrid cell. तो hybrid है मतलब it should be more than one. तो first cell अभी आपको पता चली, it is a B cell which is produced in spleen. Okay, there are different types of lymphocyte. Remember this B lymphocyte, this is produced in spleen. ओके सो ये पहला सेल आपको पता चला, हाइब्रिड सेल में से एक सेल है बी सेल्स विच आर प्रोड्यूस्ड इन स्प्लीन सेल, एंड सेकेंड जो सेल है वो है माइलोमा सेल्स, myeloma cell. So now you know कि in hybridoma technology we use hybrid cells. We use hybrid cells. Okay, my Kumar, no issue. Just listen to the lecture carefully. Okay. Good evening, NK Sandhu. Good afternoon, Ankita. Welcome, and just stay tuned to the lecture. You will get all the updates and how to tune in with science and technology. Okay, so we know hybrid cell. Now you know which two cells, from which two cells this hybrid cell is formed. First is B cell and second is myeloma cell. Okay, you know about B cell. B cell is a cell of immune system which produces antibodies. And what is the main function of hybrid cell? Antibody production. So वो काम hybrid cell में कौन करता है? B cell करते हैं. दूसरा है myeloma cells. Here अभी के लिए इतना याद रखिए, myeloma cells are nothing but the cancerous cells. Myeloma cells are cancerous cells. Here इसका मीनिंग क्या हुआ? The hybrid cell we are discussing about hybridoma in hybridoma technology. This hybrid cell is made up of a cell from immune cell, that is B cell, and a cancerous cell, that is myeloma cell. Clear? What we do through this hybridoma technology, Rekha Sharma, welcome, and just stay tuned. I will give you all the updates. Okay. Srihari, lymphocytes are the specialized cells of immune system which are mainly involved in killing or eating the bacteria or the germs. Okay, lymphocytes, they eat or kill the bacteria or germs. They are specialized cells of immune system. Now what we do, we combine the quality of antibody production of B cell. And what is the quality of cancerous cell? Tell me guys, what is the main quality of a cancer cell? I am waiting for your answer. What is the main quality of cancer cell? Okay, now see what we do. We combine the quality of antibody production of single specificity, which is the quality of B cell, as well as we combine the quality of cancerous cells to multiply. कैंसरस सेल का मेन क्वालिटी क्या होता है? ये मल्टीप्लाई होती है अनलिमिटेड टाइम, या दे do not stop multiplying, they keep multiplying, and that's why they form tumors. So cancerous cells, multiplication. B cells, yes, rapid multiplication or uncontrolled multiplication. Okay, so दो क्वालिटीज है, बी सेल की क्वालिटी एंटीबॉडी प्रोडक्शन एंड माइलोमा सेल की क्वालिटी मल्टीप्लिकेशन. सो वी कंबाइन दीज टू क्वालिटीज एंड this hybrid cell produces antibodies in large numbers. Hybrid cell produces antibodies in large numbers, and what these antibodies are? These are monoclonal antibodies. Monoclonal antibodies. Now you got it. Why in hybridoma technology we use hybrid cell? Because these hybrid cells can produce large number of monoclonal antibodies. Clear guys? If you understood, please say yes in the live chat. Now we'll go to the next point, monoclonal. I already explained you about these monoclonal antibodies. इसमें सिर्फ आपको याद कीवर्ड्स क्या क्या याद रखने? They are, first keyword याद रखना है, they are identical. These antibodies are identical. Why? Because they are produced by identical immune cell. And because they are identical मतलब हम उन्हें क्या कह सकते हैं? Clones. तो mono means single, clone means identical. So these antibodies are produced by a single or identical immune cells, and they are identical to each other. That's why monoclonal. Antibodies you already know. Antibodies you already know. Fine. Now one question for you. Which antibodies we are planning to use against SARS-CoV-2? सार्स CoV-2 के अगेंस्ट कौन सी एंटीबॉडीज यूज करना हम प्लान करने वाले? We are planning. Just answer in the comment section. Okay. Now these monoclonal antibodies, they are used for the treatment of cancer as well as they are used as anti-venom. मैंने आपको पहले ही बताया था, this anti-venom has ready-made antibodies. इसमें ready-made क्या होती है? Antibodies होती है anti-venom में. फाइन। और यहाँ पे भी हम हाइब्रिडोमा में क्या बात कर रहे हैं? टू प्रोड्यूस antibodies in large number, to produce antibodies in large number. So monoclonal antibodies, understood, they are produced by a single or identical immune cell, that is B cell, and they are clones of single parent cell. That's why monoclonal antibodies. Now how we produce hybrid cells, how we produce hybrid cells, and from these hybrid cells how we produce large number of antibodies. See Savita, mono means single, clone means identical. So these antibodies are produced by a single parent cell, that's why mono, and they are identical, that's why clonal, monoclonal. Okay, so hybrid cells, they produce large number of antibodies or large amount of antibodies. Okay, so ये ultimate aim है हमारा production of antibodies. ये अचीव करने के लिए हम exactly कैसे करते हैं? First what we do, immunization of specific animal. Immunization of specific animal, इसका मीनिंग क्या होता है? What we do, if suppose this is mice, we inject antigen, we inject antigen into this mice. Now what is antigen? या फिर सिंपल आपके अंडरस्टैंडिंग के लिए, we inject causative agent of disease, causative agent of disease in mice. Now what we do in vaccines, inactivated or attenuated vaccines में what we inject in inactivated or attenuated vaccines? Yes, tell me guys. So when we inject this antigen or causative agent of the disease in this mice, so what happens? This mice, a spleen of this mice will activate and it will start producing antibodies against this antigen. It will start producing antibodies against this antigen. Okay, so ये पहला स्टेप हुआ। इम्यूनाइजेशन का मीनिंग क्या होता है? वैक्सीनेशन। सर दे आर प्रोड्यूस्ड बाय हाइब्रिडिंग बी सेल्स एंड कैंसर सेल्स। देन व्हाई से दैट दे हैव अ सिंगल सेल पेरेंट? बिकॉज सी कैंसर सेल की हमने एक क्वालिटी ली है, कौन सी? ओनली मल्टीप्लीकेशन क्वालिटी, बट एक्चुअली उनकी जो पेरेंट सेल है, सिंगल पेरेंट सेल, दैट इज बी सेल. They are all descendant of the immune cell, that is B cell, right? Weak and killed organisms ही होते हैं ना, disease causing agent ही होते हैं ना. That's why here we are saying immunization of specific animal. So understood, immunization of specific animal, सभी को समझ में आ गया? Now next what we do, we isolate myeloma cells. Okay, and then we fuse इस एक्टिवेशन की वजह से जो बी सेल बनी है स्प्लीन में माइस के, वी फ्यूज दीज बी सेल्स विथ दी माइलोमा सेल्स. तो ये बी सेल और ये माइलोमा सेल हम फ्यूज करते हैं, तो उससे हमें क्या मिला? हाइब्रिडोमा सेल मिली. नाउ देयर आर डिफरेंट टाइप्स ऑफ सेल, जो उसमें से हम सिर्फ हाइब्रिडोमा सेल आइसोलेट करते हैं. देन वी स्क्रीन दीज हाइब्रिडोमा सेल, दैट इज वेदर दे प्रोड्यूस एंटीबॉडीज, डिजायर्ड एंटीबॉडीज और नॉट. And if they are producing desired antibodies, what we do? We use them. One by one we will go through the steps. First what we did, immunization of specific animal. I already explained you. We injected antigen into mice, then because of that antigen, spleen activates B cell which will produce spleen cells, which we are going to use with myeloma cells. These plasma cells will produce monoclonal antibodies, and now we will isolate plasma cells or B cells from this animal. तो हमें पहली सेल मिल गई हमारी B cell. Okay, next isolation of myeloma cell. Now see, These myeloma, पहला point याद रखिए, these myeloma cells are nothing but these are abnormal plasma cells which are present in bone marrow. अब यहाँ पे एक point याद रखिए, just यहाँ आ गया है तो याद रखिए, RBCs, WBC and platelet are all produced in bone marrow. These are three types of cells which are present in blood, and all these cells are produced in bone marrow. So here plasma myeloma cells are the cancerous immune cell. Myeloma cells are the cancerous immune cell which are present in bone marrow. We isolate them and then we फ्यूज दीज माइलोमा सेल्स विथ बी सेल्स और उससे हमें क्या मिलेगी, हाइब्रिड सेल, एंड वी यूज हाइब्रिड सेल, दैट्स व्हाई दिस इज हाइब्रिडोमा टेक्नोलॉजी. क्लियर? सो अब बहुत ही इंपॉर्टेंट है, एक पॉइंट आपको हमेशा के लिए याद रखना है, myeloma cells are immortal, means they never die. Myeloma cells are immortal, they never die, and because these cells are cancerous, so they have multiplication property as well. Multiplication and immortality, ये दोनों क्वालिटीज है. जैसे यहां मैं आपको बताता हूं, व्हाट इज द लाइफ स्पैन ऑफ आरबीसी, कैन एनीवन टेल? हेलो गाइस, बताइए आरबीसी की लाइफ स्पैन क्या है? ठीक है, ये इमॉर्टल वाला पॉइंट याद रखिए. सर, इज माइलोमा सेल्स एंड ल्यूकेमिक सेल्स आर सेम? नो श्रीहरि, माइलोमा सेल्स आर इन बोन मैरो एंड ल्यूकेमिया दीज आर द सेल्स, कैंसरस सेल्स इन ब्लड इटसेल्फ. ठीक है, व्हाइट ब्लड सेल्स की अगर एक्स्ट्रा ग्रोथ होती है या कैंसर होता है तो उसे हम ल्यूकेमिया कहते हैं. सो ल्यूकेमिक सेल्स आर इन ब्लड एंड माइलोमा सेल्स आर इन बोन मैरो. फाइन. नाउ अब हमें क्या हुआ, नाउ यू नो व्हाट वी गॉट बी सेल्स और वी गॉट plasma cells. Yes, very good. 120 days. No, Pashupanti Balaji, it is not 72 days, it is 120 days, and it was a single liner asked by UPSC. Okay, 120 days, correct. So we have B cells. From where we got these B cells? By immunization of specific animal. Okay, by immunization of specific animal, that is mice. Then we have myeloma cells. From where we got these myeloma cells? We isolated that from bone marrow, and now we will fuse these two cells by, there are different ways. Either we can fuse them with polyethylene glycol or electrofusion. Now here I want to tell you guys, PEG medium or electrofusion, this is not important for us. UPSC will not ask this. It is just for your understanding कि बी सेल और माइलोमा सेल को फ्यूज करके उनसे हाइब्रिड सेल कैसे बनाते हैं. तो बाय दिस मेथड, आइदर पॉलीइथाइलिन ग्लाइकोल मीडियम और electrofusion method. These are the two methods for fusion. So once this fusion is complete, we get 5 different types of cell in that medium. We get 5 different types of cells. Savita, only 120 days. Okay, so first is fused plasma cell, no use for us. Fused myeloma cell, no use for us. Unfused plasma, no use. Unfused myeloma, no use. So we got hybridoma or hybrid cells which we will use. We got hybridoma or hybrid cells which we will use. Clear? So fusion is done. Rinki Panwar, only 120 days. Yes, Aarti Gupta, you will get PDF of this slides. What you need to do, you need to subscribe to my Telegram channel. The name of my link, or you can search me from like this, Sachin underscore Kadam 1, t.m/Sachin underscore Kadam 1. This is my Telegram channel. Here you will get all the notes in PDF format. Okay, Aarti? Fine. Now, what happens here, guys? Don't go in technicalities. Here, यहाँ पे पहला पॉइंट क्या कह रहा है कि बिफोर मल्टीप्लीकेशन ऑफ एंटीबॉडी दीज सेल्स नीड टू कॉपी डीएनए, क्योंकि ये हाइब्रिड सेल है तो इसका खुद का एक डीएनए होना चाहिए. दिस सेल नीड टू मल्टीप्लाई एंड मेक DNAs, and for DNA formation we need nucleotide, and these nucleotides are provided by HGPRT enzyme. HGPRT, hypoxanthine guanine phosphoribosyl transferase. So this enzyme is secreted only by hybrid cells. That's why we use hybrid cell. सो टेक्निकलिटीज में इस स्लाइड में हमें कुछ भी इंपॉर्टेंट नहीं है, ठीक है। आई एम जस्ट एक्सप्लेनिंग इट एज स्टेप बाय स्टेप. So what we did, we isolated B cell, we isolated myeloma cell, we fused them, and now we will start multiplication of these hybrid cells to produce monoclonal antibodies. Fine. Now for multiplication we use two methods. Everyone, those who are from science background, everyone knows in vivo means actual in the living organism. When we multiply any bacteria, virus or cell in actual living organism, so that is known as in vivo. In vivo में हम क्या करते हैं, we inject hybrid cells or hybridoma cells in the animal, and now these cells will multiply in the body of this animal. And as you know, these cells have two qualities. They can produce, what are the two qualities of these cells? They can produce antibody as well as they can multiply. So because of these two combining qualities, they produce large number of antibodies in that animal, large number of antibodies in that animal, and our aim is achieved. So it can be done by in vivo, that is in animal, or in vitro. What we can do, we will culture or we will grow these hybrid cells in nutrient rich medium, which is normally science student, nutrient rich medium, which is mainly used for culture. Please tell me, mainly used for culture. Ching Jongske, I am from science background. It's kind of revising everything. Thank you, sir. Okay, no issue. Ching Jongske. Just remember, hybridoma technology is important for your preliminary examination. Okay, so we use agar as a nutrient medium. Most common, it is extracted from algae. So we grow these cells in petri dish in this agar, and these cells multiply, and again these cells while multiplying they produce large number of antibodies which we need, which are monoclonal antibodies. Fine. So last point, for again for your prelims, क्या पूछ सकती है यूपीएससी प्रीलिम्स में कि which of the following are the applications of hybridoma technology? You know only one application through this lecture, that is to produce monoclonal antibodies, that is for development of immunity, that is to fight diseases. But hybridoma technology is also used in the identification of ABO blood group, because ABO में भी क्या होता है, antibodies ही role play करती है. We will take a separate lecture on blood groups, so in that I will explain you this. So this is used. Then detection of pregnancy. Here remember, the first reaction of human body to pregnancy is, बॉडी कंसिडर करती है इट इज अ कैंसर. इनिशियली क्या लगता है बॉडी को, ये एक्स्ट्रा ग्रोथ है, तो बॉडी एचसीजी ह्यूमन कोरियानो गोनाडोट्रोपिक हार्मोन रिलीज करती है, विच इज रिलीज्ड बाय कैंसर ट्यूमर्स एज वेल एज ड्यूरिंग प्रेगनेंसी. सो टू डिटेक्ट pregnancy, hybridoma technology is used. Then as we know, we use this technology for production of monoclonal antibodies. So it is used to for separation of one substance from very similar molecule. Okay, then it is also used in the purification of individual interferon from monoclonal antibodies, as well as in activation of T lymphocyte which are killing the transplanted organs. So these are the applications of hybridoma technology, and it is also used in treatment. Therapy में भी use करते हैं. Fine. So my advice to you, if you come across, I'm just writing down the words. If you come across these words in the Hindu, just read it carefully and try to understand. Hybridoma technology, monoclonal antibodies. Okay, then if you come across B cells, so just read them properly. Okay, so this is all about hybridoma technology. Okay guys, so subscribe to Unacademy's YouTube channel, Let's Crack UPSC CSE. Hit the bell icon so that you will get regular updates of videos and you will not miss any video. And subscribe to Unacademy learning platform or learning app. Use my code SQS, you will get immediate 10% discount. So thank you very much guys. Now those students who were saying they are new, their first lecture for them, I'm conducting lectures of science and technology every day at 8 a.m. in the morning on Unacademy Learning app. For that, you need to download Unacademy Learning app and follow me. That is Sachin Umrao Kadam. Then you will get regular updates. And second, I'm conducting regular lectures on current affairs from The Hindu 11 a.m. every day on YouTube. So stay tuned as well as watch my previous videos. So your fear of science and technology for UPSC CSE will be over. Okay, guys. So thank you very much. See you soon. Let's meet tomorrow. Thank you. Bye bye. Take care.